Morning. Okay. Okay. There we go. All right. So check in on iClicker. Don't forget to check in on iClicker. Yes, I imagine they are. I imagine they are. Oh, an exam. Exams are back. On the back. I think yours, I had to fit yours in. Because it was nice. Yeah. It should be there. Should all be there. It's a 4-point curve. So whatever you have, a 4-point curve, but I don't have the grades posted to Moodle yet.
I was literally checking like 30 minutes ago.
What you have plus 4.
So this
Whatever your
doesn't
percentage
factor
is.
into the curve.
Correct.
Okay.
Because when I grade them individually,
You grade them exact as they are.
right.
Gotcha.
And then whatever grade you have, plus 4.
Okay, thank you.
Yes. It's a meeting now. Just a meeting.
I was gonna say, I have a bunch of them.
No.
Whatever. They don't come for points, so.
I don't, but
But I mean, I know I was there, is what I'm saying.
right. All right. So I thought on the exam, while we're on the topic of exam, I thought it was better to have more points and one less question. How did y'all
I actually
feel
really
about
like
that?
this exam.
What?
I was gonna say, I like this exam, but I gotta see my grade first to know for sure if I like it or
If you
not.
really like the exam, well, you know, it is what it is. Okay. So we have another exam on, what do we have another exam? Gotta pull that up. Let's see. I pulled up Moodle over here. When's our next one? Here we go. Class calendar. Almost couldn't find my own class calendar. All right, so drop day is next Tuesday. Then we have on Thursday our case study number 4. And then we have exam 3 on Tuesday. Okay, so we have today and tomorrow to finish, today and tomorrow. No, where are we? We're here. Today, Tuesday, and Thursday to finish 18. Then we'll have our case study, and then we'll have our exam, and then we'll do chapter 19. It'll be awesome. And then we'll have our exam. When's our final? Our final's not till Tuesday. That's great. That's great. So that's right, because that's why I made the chapter 19 homework not due till Monday. We are going to have 2 case studies on this exam. That is correct. There'll be 2 case studies. It could be. They are going to be on the exam. Yes, they are going to be on the exam. All right. So we ready to switch back? Let's see if I can do this full screen. Okay. All right. So good. Questions? Any other questions before we get rolling on content? All right. So we're talking purine and pyrimidine biosynthesis, right? So last time we talked about not biosynthesis, but what kind of pathways? The salvage pathways, right? Because it's cheaper energetically to salvage what you can rather than synthesize it from new. And we'll kind of keep track of the ATPs as we go through these synthesis pathways. So the differences between purine and pyrimidine biosynthesis really are that purines, you build on the ribose sugar, whereas pyrimidines, you build the base first, then you attach the sugar. So they kind of go in reverse of one another. So we'll start with purines and then we'll do pyrimidines. But our purines, what you're going to do is you're going to actually make an intermediate that is not, what are our purines? Let's do that. What are our purines? Pure as gold. Pure as gold.
Oh,
A and G.
I don't even want.
Yeah. So what you're going to do is you're going to make a common intermediate that is inosine 5 monophosphate. And then from inosine, we're going to make adenine and guanine. And so if you want to get technical about what are your purines, what are the purine bases, right? We said A and G in the previous lesson, but do you remember how I said there's going to be another one? So your other one is hypoxanthine. So if you want to go up and you want to add that right here, right? Hypoxanthine. That's going to be an additional purine. Okay. All right. So coming back. So it's kind of built in 2 stages because you have 2 rings, right? The purines have 2 rings. The pyrimidines have 1. Okay. So if we kind of just side by side do a comparison, what's the difference between purine biosynthesis and what's the difference between that and pyrimidine? So in purine, the sugar that we're going to start with is Prpp, right? So technically, if you're counting how much ATP you're going to have to consume in order to make this stuff from scratch, you have to count the ATP that's required to make Prpp, right? Because you want to be able to attach things to the ribose sugar. Which carbon is the base attached to in Prpp? Like if we go back up, if I'm going to make, here's Prpp, right? If I'm going to make any given nucleotide, which carbon? It's attached to carbon 1. So in Prpp, which carbon is going to have a pyrophosphate? Carbon 1. Because we want to energetically drive the reaction. And so if we put the inorganic phosphate, pyrophosphate there, we'll able to swap out the pyrophosphate for the base and use the cleavage of pyrophosphate to drive the reaction, right? Okay, so here we were. So for purine, we're just talking about purine first. We're going to start with the sugar. We're going to build the base directly on the ribose. So here's hypoxanthine. This is going to be our intermediate base that we're going to form. How is the structure of hypoxanthine similar and different to adenine and guanine? Go and look. What's the difference? If I wanted to make adenine, how do I change hypoxanthine? Go look in your structure, take a second. So get rid of the hydrogen, add a double bond. It's a terrible double bond, but okay, and there's something else. And H2. Okay, so that's adenine. All right. So now what if I want to convert that to, I'm going to erase this. Y'all got this? Yeah. Okay, I'm going to erase it. So then I want you to convert it to guanine. What are you going to have to do? It's even easier for guanine. Where?
On the
Third carbon.
Why do you need to add an H? Where? Yeah, that's all. That's it. Really easy, right? So you have to think that it's really easy to get the carbon components of all of this stuff, right? But what's in these bases that we kind of have to keep track of where we're getting things from? So those nitrogens. So as we look at these pathways, you're going to see really some commonalities. And when we talked about nitrogen metabolism and where all the nitrogens were coming from, you're going to get to see that. All right. So we have our hypoxanthine. We can take hypoxanthine. It's attached to ribose. So if we have hypoxanthine attached to our ribose, we call that inosine monophosphate. So this whole thing is inosine. The red is hypoxanthine. Okay. So we can take inosine monophosphate and then through multiple reactions, either go to AMP or go to GMP. Okay. All right. So it's going to be a little different for pyrimidine. For pyrimidine, what are we making for pyrimidine? What are our pyrimidines? C, T, and U. Okay. So our pyrimidine biosynthesis, what we're going to do is we are going to do the opposite. We're going to build the base, and then we're going to add the ribose sugar to it. So remember how I said you had an additional base to learn for purines? Well, you have an additional base to learn for pyrimidines. So orotate is the pyrimidine base that you need to go up and add to your slide up here. There's orotate. Let's see. Oop. Here. Orotate, not orotate. Okay. So you have an additional base to learn for each. Yeah. So these are the intermediates that are required for biosynthesis. Okay. So in this case, we make our ring first. Then what we're going to do is we're going to add our ring to PRPP. But again, you're going to add it to the same carbon. You're going to add it to carbon number 1. And then you make orotate monophosphate. And then from that, you make UMP. And then UMP can be converted to CMP, TMP. Okay. All right. So we're going to tackle the purine first. The purine, kind of a little easier, I think, to keep track of. So one thing that you kind of have to, well, a few things that we kind of have to keep track of as we look at these biosynthesis pathways is we want to look at ATP that we're consuming. And we also want to kind of think about regulation. So we're going to keep coming back to those two topics over and over again. All right. So the sugar that we make, if we're thinking about making it from scratch, where are we getting ribose 5-phosphate? Pentose phosphate pathway, right? Then we're going to go through Prpp synthase and we're going to make Prpp. So we've already had to invest one ATP, or look what you're doing. You're going ATP to a MP. So this is really like 2 ATP equivalents. Right? So kind of keep yourself a running total going on the side. All right. So now the next step that we want to do is we want to start adding some nitrogen groups, right? Because all of our bases have lots of nitrogen. So our first nitrogen is going to be obtained from glutamine. So that enzyme is glutamine Prpp Amidotransferase. So that's exactly what it sounds like. We're going to take glutamine, we're going to pull out the nitrogen, attach it to carbon 1, and we're left with glutamate and inorganic phosphate. So those will cleave and we'll make 2 inorganic phosphates. Okay. These 2 enzymes, Prpp synthase and glutamine Prpp amidotransferase, these 2 are very, very highly regulated by feedback inhibition. So what is feedback inhibition specifically?
When you make enough your product.
Your final product to come back and inhibit the beginning, right? So that's exactly what we want, the final to come back to the beginning. So we'll,
inhibits Purine Synthase and 5-phosphoribosilamine inhibits glutamine Purine Amidotransferase.
and we'll talk in lots of detail about regulation, but just remember, you kind of really want to regulate those beginning enzymes. All right, so now we have one nitrogen attached. We're going to be attaching other nitrogen groups, right? So the next nitrogen group comes from glycine. Now this nitrogen group coming from glycine comes at the cost of 1 ATP. So you're keeping track of this, right? Keeping track of those ATPs. All right, so we've added our second nitrogen group. And do you have to know all the names of the intermediates and stuff like that? No, do not memorize the intermediates. Do not memorize intermediates. All right, so the next one comes, our next group is going to come from informal THF. This gives us our formal group. So this is our formal group right here. And this one, right, comes at no ATP cost. But our next one is glutamine. We're going to do another nitrogen here. So how much ATP are we using up? One more ATP, right? Because we're going to ADP. So we're only costing 1. Okay. So we get our third nitrogen that replaces the oxygen in the carbonyl there, right? Now we're getting close to being able to close our ring. So at the cost of 1 more ATP, what we're going to do is we're going to close that ring from here to here. That's the ring that we're going to close. And you can see right there, we have officially closed our ring. So now are we done? No, because this is a purine. So we've only made 1 ring. So this is stage 1. At stage 1, we have 1 completed ring. So at the end of stage 1, how much did stage 1 cost us? Stage 1 has cost us 5 equivalents of the ATP. Do you need to know your nitrogen sources though? Yes, you need to know your nitrogen sources. Know your nitrogen sources. Okay, so now we're going to go on to stage 2. At stage 2, we start and we already have 1 ring completed. So now we're going to start building onto our other ring. We first get carbon group from bicarb. So what's that? At a cost of 1 ATP. At a cost of 1 ATP, because we're trying to keep track of that. The next nitrogen that we're going to get, because we're not going to keep all the stuff from aspartate. Actually, the only thing that we're going to keep is our nitrogen group right there. Comes again at the cost of an ATP. So we're going to cut off all of the excess stuff that we got from aspartate, release it as fumarate, and now we have our nitrogen by itself. So that doesn't cost us an ATP. We're going to again get another formal group to attach to the ring. So now we're really close to having a completed second ring. We're going to dehydrate and complete our second ring. So now we have inosine 5 monophosphate. At a cost, so stage 2 cost us 2 ATP. So stage 1 and 2 together cost us 7 ATP. Whoa. Whoa. 7 ATP. All right. So this is why salvage is so important. A cost of 7 ATP to make 1 is really, really high. So you want to salvage as much as possible, right? the cell is really great at recycling. So all of these reactions occur differently in eukaryotes as prokaryotes. So eukaryotes actually have 10 different enzymes that they use to make one purine mononucleotide, but eukaryotes only use 6. So there's a little more repetition in bacteria. All right, so now that we have inosine monophosphate, so now we're not drawing the sugar anymore, we're just putting the words there, right? Y'all see that? Okay, so we have inosine monophosphate. We want to make from inosine monophosphate 2 different nucleotides, right? What do we want to make? GMP and AMP. Okay, when you are looking at these, what you're going to notice is that you're no longer investing just ATP. And this gets really important. What are you investing? GTP. I'm going to invest GTP to make, what's my final product, right? Here's my GTP investment. What am I making at the end? AMP. Okay, if I want to make GMP, what am I going to invest? ATP. This alternating investment versus product that you're making, make sure that you don't deplete one and not the other. Okay, so that's kind of an important little thing to note. Okay, so remember we said what are the differences, right? What do we have to do? In order to go to ATP, we're going to have to add another nitrogen group. That nitrogen group is going to come from aspartate at a cost, right? So you guys are totaling this cost in your mind, right? So now we're up to 8 ATP, all right? Then we're going to cut off the extra stuff that we didn't need, release it as fumarate, and now we have AMP, right? At a cost of 8 ATP. 8 ATP equivalents, right? Not really 8 ATP, but 8 ATP equivalents. So now if I want to go and make GTP, I need to dehydrate, right? I'm going to make xanthose 5-monophosphate. Then I need another nitrogen group, right? Both of them needed an extra nitrogen group. So at an investment of 1 ATP, we're going to use glutamine. We're going to use one of glutamine's nitrogens. We'll release glutamate, and we will make GMP. Okay. So when you think about this, right, it's really significant that we want GTP used in order to drive AMP synthesis. And so we're going to see that in lots of different regulation. So now we have our monophosphates, but can we add monophosphates to a growing mRNA, DNA chain? What do we need? We need a triphosphate. We always need a triphosphate. So go back to your salvage pathway, and you can see at the very top, when you finally got back to the very top, you had to add a triphosphate. So are we even done with the ATP investment? No. All right. So now, depends on what you're starting with. On the left, we're going to start with AMP. So we have AMP. We're going to invest in ATP and get out an ADP, and we're going to convert that phosphate group that's on carbon what? Not carbon 1. Carbon 1 has the base. Carbon 5. Carbon 5. So we're going to add our phosphate, right? Now, when we do this, that first addition requires ATP. The second addition requires any nucleotide triphosphate. Does not matter which one, any one will do to add that second phosphate. So now that I have a triphosphate. So now we can go in and use this for either DNA or RNA synthesis. It depends, right? So if I want to do it for DNA synthesis, what do I have to do? I have to lose Where am I losing this? It shows it just as water coming off, but where am I losing it? I'm losing it on the second carbon. So this water coming off is actually from here. Right? Because it's deoxy. But then you do the same thing. You use up some nucleotide triphosphate, doesn't matter which one, and then you're making DATP, and you can use it for ATP synthesis. Okay? So the enzymes that phosphorylate, right, the nucleoside diphosphate kinase, these are similar in the salvage pathway, right? Same thing, nucleoside diphosphate kinase, same enzymes, right? This one's just different. You don't have this one in the salvage pathway. Basically, you're salvaging either RNA or you're salvaging DNA. So you keep them kind of separate. All right, so if you have guanine, it's very similar. Instead of having adenylate kinase, what do we have? Guanylate kinase. Again, it's an investment of ATP in the first stage. Ooh, ATP in the first stage, and then any given nucleotide in the second stage. And you do that same thing. If we want to make it for RNA, we don't have to do anything additional, but if we want to make it for DNA, we have to reduce it to get rid of that oxygen on C2. Yeah. Not too bad, huh? So from that, we have either ATP or GTP or DATP or DGTP. Cool? Okay.
Does that make the total ATP 9?
That makes the total ATP 9. Yes, that makes total ATP 9. From start to finish. Yeah, 9, we'll put equivalence behind it. Because it's going to depend on which one you're making, what you are, which DNTPs you're using, or NTPs just in general. Okay, so we know in general that feedback inhibition will ensure that you get the correct proportion of each of our different nucleotides, right? It's the same kind of thing that we talked about. Remember when we talked about amino acid synthesis? Do you remember how we had all that feedback regulation in amino acid synthesis? It's the same thing in nucleotide synthesis. So if you kind of look just generally at this pathway, Prpp synthetase is a big site of regulation. So if we look at Prpp synthetase, it is going to be inhibited by ADP. It is also inhibited by GDP, right? So Prpp synthase, you can kind of start yourself a list. Prpp synthetase, TH. That's why, yeah. Synthetase. So you've got inhibition by ADP, that's here. You have inhibition by GDP, that's here. Okay, so why do we want this inhibition so early on? It's very costly. So we want to stop the whole process from the very beginning. Where's the first place that we have an ATP investment? In Prpp synthetase. Requires not 1, but 2 ATPs. So we want to stop that as early as possible, right? Okay, so the next one, we said the glutamine Prpp amidotransferase. This one is inhibited. Look at this one. So 2, we're going to say glutamine Prpp transferase. We'll just call it that, transferase. So this is inhibited by all, right? ATP, ADP, AMP, GTP, GDP, GMP, by all. We'll just say NTP, DP, MP, right? By everybody. Well, not everybody. We'll say all purine. That's our next ATP investment. Now we go through lots of reactions, right? And we make our intermediate IMP. So our intermediate, this is a point where we need another set of regulation. Why do we need another set of regulation at the point where we have IMP? Like, yes, I know we want to inhibit the beginning because that will prevent excess general purines being made. But why do I want to have inhibition immediately after IMP?
That's how you select which one.
That's how you select, do I want to make ATP or do I want to make GTP? Okay, so 3 is after IMP, right? And this allows us to be selective. So if we have a lot of AMP is going to come back up and negatively regulate ATP formation. If we have a lot of GMP, we're going to prevent GTP from occurring. All right. So this step in the regulation, you get independent regulation of TP, independent regulation of ATP versus GTP.
For its own pathway. Like the GTP only works to inhibit the creation of CMP.
It's not only, be careful with that because it also inhibits up here.
Well, I mean, like,
So it inhibits the whole pathway also. So for this point, why do you want GMP to inhibit the conversion of IMP into this side of the pathway? Because if you have lots of GMP, but you don't have enough AMP or ATP really.
then you can stop that one and force IMP to go into adenyl succinate and then go
Yep.
into the ATP pathway.
Yep. So you want to be able to decide, do you want to make purines in general? And do you want to make specifically ATP or specifically GTP? So that gives us our balance in our two pathways. All right. So before we go and talk about synthesis of pyrimidines, I want to know what's going to happen if I have excess purines. Like what happens if I just have a lot of them? I've broken down, I don't know, maybe a lot of RNA, and I have a buildup of purines. How do you get rid of it? You're going to make what's called uric acid. So if we look, uric acid is eventually, right, we've got a lot of nitrogens, is going to eventually go through a nitrogen degradation pathway. So if we look at AMP, right, we've got AMP, we're going to make, from AMP, we're going to cut off our ribose, right, and we're going to get adenosine. So once we have adenosine, now we can start pulling off nitrogens. We can use a deaminase and we can pull off nitrogens. Do we know how to get rid of nitrogens in the body in this free form? Yes, we do. Yes, we do. Yeah, we're going to eventually get to the urea pathway, right? Right? Exactly. Okay. So if we go this way, right? What we're going to do is we're going to cut off the sugar and we're going to have the base. Now here's the sugar by itself. Once we have the sugar, ribose 5-phosphate, right? What we can do is convert that to inosine. So once we have inosine, we can convert it back to hypoxanthine, and then eventually xanthine to uric acid. The great thing about uric acid, what's the point of changing it into uric acid? Uric acid is soluble. I'm sorry, it's insoluble. It's insoluble. So a lot of times what you're going to see, you know, like birds, don't poop. They don't pee. Their pee is in their poop, right? So when they're excreting all that, you're washing it off of your car and that kind of stuff, there's lots of uric acid inside of their excrement, right? And so that's what gives it the great smell and everything, which is awesome. Well, that's what about nitrogen. Nitrogen stinks. That's what it is. All right, so you have a very similar pathway if you were going to start out with GMP, right? If we start out with GMP, we go through a nucleotidase, and what do we make? Guanosine, right? And from that, we can convert that guanosine into the same guanine, and then to xanthine, and then back to uric acid.
So why does the adenosine have to turn to inosine, but guanosine doesn't?
It's just the enzymes that we have. It's the most efficient way to do it. It's the most efficient way to do it.
Okay.
Yep. Okay. All right. So anybody know what's the problem with uric acid? Right? If we have uric acid, we can take that uric acid and we can break it down, right? And different organisms will break it down into different other byproducts and things like that. But if we lack, or the enzymes that are used to start breaking down this uric acid don't work as well as they are supposed to, what disorder are humans going to have?
Yep.
They're going to have gout. They are going to have gout. Have you ever seen, I should have brought a picture. Have you ever seen what actual uric acid crystals look like? They look like needles with like really sharp ends on either side. Did I not? Oh yeah, there you go. There you go. Exactly. Exactly. So they look like super, super sharp needles, right? So this is what happens. You have an excess of uric acid. It's insoluble. And when you have a buildup of it, right, it's a solid crystal in this form. And so it's actually going to get pulled down by gravity. So people who complain of gout, where is the site of in their extremities, and lots in their feet? Their big toe is the worst one, right? The big toe is like the worst one. But you have all these crystals that get trapped in really small blood vessels, and it's extremely, extremely painful. Okay, so we talked about this. All right. So if you don't degrade purines the right way, you're going to get a buildup of uric acid. Uric acid builds up not only in the joints, but also the kidneys, right? And so you end up having too much of things floating around in the bloodstream. So if you talk to anybody who has gout, right? If they stub their toe and they break open those blood vessels and you have all those really, really sharp needles, you're going to hear that people who have gout, like, it's extremely painful when you stub your toe, like extremely painful. My uncle had gout. He had the gout. That's what he called it, the gout. I think that's a Kinase thing. What does she call it? The Google? Why the Google?
I don't know. That's just how she always talks about it.
That's how grandparents do, right?
Yeah, I think that's just a grandparent thing.
So there are a couple of different ways that you could have deficiencies in order to have gout. Sometimes you're going to have gout if you have a deficiency in the breakdown pathway, and there are other deficiencies that we're going to talk about. So the first one that we talked about is gout. There are a couple of different ways that you can have gout. If you have your ribose 5-phosphate, right, you go to PRPP. If you lack this glutamine PRPP amidotransferase, you're not going to be able to utilize the PRPP. So now you're going to be insensitive to feedback inhibition, and you're going to have gout. The other way that you have gout is that buildup of uric acid gives you gout. Either way, you're going to end up with the same overall product of having an excess of uric acid, because you're going to continue through here. If you don't have feedback inhibition at PRPP amidotransferase, this is just going to keep going. There's going to be no, hey, we've made enough, stop. It's just going to keep making purines. You make too many purines, what are you going to get? An accumulation of uric acid, which is going to give you gout, right? Okay. So how do you treat patients who have gout? You do tell them to change their diet. Stop eating red meat. Why stop eating red meat? It has a lot of protein. So if you restrict your diet of purine rich foods, right? Red meat, organ meat, all those kinds of things, then you can have less that you are ingesting. So then you have less overall, right? So you'll only get it from the synthesis pathway. You can also give a drug, and the drug is a competitive inhibitor of the xanthine oxidase, which is one of the enzymes in there, but you don't have to know that. But there is a drug that you can give, but it's not perfect. It's not awesome. Okay. So there's another really big issue here. When we try to convert adenosine to inosine to then be able to degrade it, right? Or, what is SCID? I've heard that before. It's an immunodeficiency. And so if you can't convert adenosine to inosine, inosine is, remember, our intermediate, right? And we can take an inosine and we can convert it to either adenosine or what? Guanine. So if you cannot make enough guanine, when you go to replicate your DNA in highly rapidly dividing cells like your immune cells, you're going to have issues. So you have a huge suppression in the immune system. And if you have a huge suppression in the immune system, a couple of different things can happen. You can have an inability to fight off infection, but what else does your immune system do for you that we talked about in the very beginning of this chapter? It screens your body for what on a daily basis? Cancer. So what you see in people with these immunodeficiencies like this is that not only are they more susceptible to infections, but they're also more susceptible to cancer. So it's really pretty serious. No, MS is different. No, MS is a hyperactive immune system towards the myelin sheath that covers the nerve cells in your brain. And so your immune system will break down that myelin sheath and stop the communications within the brain.
So this is hypoactive.
So no, this is, no. That's hypoactive. Oh, I'm sorry, that's hyperactive. This is hypo. You are correct. That is hypo. You are correct. You are correct. Okay, and then the last one, I don't even know how to pronounce that one, so I'm not even going to try. So when we make hypoxanthine, remember that your body is all about recycling, right? So at this hypoxanthine stage, what we can do is we can take this back to either IMP, or in the presence of guanine, we can take it back to GMP, right? So if this enzyme is inhibited, you get, and I'm not even going to try it. I'm not even. There are some that I'm just like, why? You had to name it after somebody. Why did you have to name it after somebody? It's very rare, very uncommon, but it's a lot of really weird neurological symptoms. And I don't know the biochemical explanation for exactly why. I haven't done a whole lot of research on this one, but it is recessive, and the gene is located on the X chromosome. So it's X-linked and it's recessive, but it's a really weird, but it might be like, if you don't have enough of the individual nucleotides during development, right? Then your brain doesn't develop the way it's supposed to. leading to not cognitive defects, but like social defects, right? Because it's anxiety, it's self-mutilation. It's really strange. It's really strange. Okay, so the last one, this is the one that we were talking about, the SCID. So adenosine deaminase deficiency, not deficiency. Haha. So this is severe combined immunodeficiency disease, right? So this prevents you from making your nucleotides. And when you don't make the nucleotides, the big problem is on your immune system. And the most rapidly replicating cells in the immune system are going to be the B and T cells. So if you can't make enough DNA for your B and T cells, there's really nothing that you can do to treat that. So what you have to do is a bone marrow transplant to replace those cells so that they have new DNA in order to make all the nucleotides to make functional B and T cells, right? All right, so if you have somebody who has severe combined immunodeficiency, they've done movies and stuff like this. This is like the bubble kids, right? That they have to live in complete like isolation, right? This is the deficiency for that. Like, I think it's kind of cool when, you know, you've seen stuff like that in the news and that kind of stuff, and you're able to put an actual biochemical defect to name to the system, you know, and understand why. And why are they in a bubble? Because you're trying to prevent not cancer, right? You can't control that from the bubble. You're preventing infection issues. Yeah. So one thing that they're trying to do for individuals with SCID is that they're trying to do gene therapy. So what is gene therapy? Well, I'd really like to know if they're changing this procedure now that we have CRISPR. I'd really love to know that, but I don't. So first, you're going to take a virus, right? Now, viruses replicate differently. Every virus has a unique way that it replicates. The viruses in particular that they use for gene therapy are called retroviruses. So what do retroviruses do? Anybody take virology? They go in, well, what does that mean? They go in reverse. They go RNA to DNA, and then they insert their DNA into your DNA, right? So now you're carrying around a little bit of viral DNA in your own genome. Well, yeah. So how can we use that? That sounds really dangerous, right? And it really sort of is. And especially in the very beginning, because you don't know where the insert is going to happen in the genome. So you take this virus and you mutate the virus so that it's no longer going to make more viral particles, right? It's only able to copy one portion of the DNA, and that portion of the DNA contains the enzyme, the correct enzyme, a good copy of the adenosine deaminase gene. Takes that good copy, infects the cell, integrates that good copy into the patient cells. But you can't just inject this virus into the patient, because where's the virus going to go? Like everywhere, right? So before you can give it to the patient, you actually have to harvest the correct cell type from the patient, then infect the cells in a petri dish, allow them to grow. Now, is every single cell going to be infected by the virus? No. So you have to have a way to select out the ones that didn't get the insertion, propagate them, grow them, and then put them back into the patient. And so the big issue with this was that in the very beginning, we didn't have a targeting mechanism. And so if you insert this into a gene that is supposed to, let's say, control cell cycle, right? Let's say it's a gene that's there to put the brakes on the cell cycle when it needs to stop. If you disrupt that gene, what have you now done? You've given them cancer. So maybe you treated They're skid and you just gave them cancer. So you have to be very, very careful about that. I mean, it's great. It's great. And they've hugely, vastly improved the targeting mechanism that they use. But it's something that you kind of have to keep in mind, right? Pretty cool though, right? I could talk about that kind of stuff for days and days and days. All right, so now we're gonna move on to pyrimidines, right? So what's the difference in pyrimidines overall? We're gonna, well, they only have one ring. We're gonna build the ring, then we're gonna attach it to PRPP, right? How do purines work? You build it from the ring. The first step is to attach a nitrogen to the PRPP, right? So in this case, we're not going to talk about PRPP until our ring is made. So it's a little bit different, but it makes sense, right? Because you only have one ring. You're not trying to build a whole lot. Okay, so I thought this was a nice little thing that they had because it shows you where each of the atoms in the ring come from, right? But we're gonna take it step by step. So when we make pyrimidines, remember when we talked about in the very beginning, you're gonna make a common nucleotide, right? What's the common nucleotide? Uracil monophosphate. So we're gonna start to make our ring, we're going to start with bicarbonate and we're going to start with glutamine. We're going to combine the two and we're going to make carbamyl phosphate. Where have we seen that before? In the urea cycle, right? Okay, so do we have an ATP investment? Yes, how much? 2. Okay, 2. So now, we're going to go through aspartate trans carbomylase. So this is going to attach a whole lot to our group. We're going to attach it from aspartate. We're going to get almost the rest of the ring this way. The only other thing that we need to do is to attach the carbons that we got from aspartate to our nitrogen, right? So this is really a whole lot easier of a synthesis. It's really only one ring. Right? So we go through a dehydration reaction. We link the carbons that we got from aspartate to the nitrogen that we got. Where do we get our nitrogen from? Glutamine, from our nitrogen from glutamine. We go through a dehydrogenase reaction and we make orotate. Remember, orotate was that additional base that we were talking about? This is the extra base that you had to know. So now that we make orotate, we can go and from that attach that orotate to the sugar. Once we attach orotate to the sugar, we're going to make orotein 5 monophosphate, right? Because it's going to be attached to the first carbon. Now what are we going to do? Convert that to uridine monophosphate. Okay, so how many ATP investments did we have here? 2. We didn't have very much investment on this one, but does that make sense? You're only making one ring and you're starting out with a whole lot less substrates in order to make that one ring than we did in our purines. Okay, so now we have UMP. We can't use monophosphates to do anything. So what are we going to do? Phosphorylate them, right? So we have UMP kinase. We're going to add a phosphate group. But did we do an investment there? Yes, we have an ATP investment there. Now we're going to have to add another phosphate. There's another investment. From there, we can make UTP. Now, what was the difference between uracil and cytosine? Instead of having
Instead of
a
an
carbonyl,
H2, or instead of that carbonyl, it was an H2.
it was an NH2. That's it. So you lose the hydrogen here, you make the double bond, and you convert that oxygen to a nitrogen. That's it. So it does take an investment of ATP in order to make that work. Okay. Not too bad, right? Good. Okay, so now we talked about regulation of purines. Now we're going to talk about regulation of pyrimidines. So just like we talked about before, we're going to have feedback inhibition, but we're also going to have allosteric activation. So when you think about what sort of molecules should activate this pathway, when do you want to make nucleotides? You have to have enough substrate, right? So one of the substrates is the sugar. So because this sugar comes in at the end, the sugar is going to be a positive allosteric regulator for purine biosynthesis. Then we also have ATP, because do you really want the cell replicating if you have low ATP levels? No, not really. So you're not going to make more nucleotides if you don't have enough ATP. Then you really need to go through the salvage pathway. And then your negative allosteric regulators, UDP, UTP, and CTP. So those are all of our feedback inhibitors, are all endpoints that are going to come back up and regulate the beginning. So now the regulation is slightly different in bacteria than it is in animals. So on the top, this is bacteria. On the bottom, this is animals, right? Looks a little more complicated for animals, doesn't it? Okay, so if you remember back in, oh gosh, I don't remember what chapter that was in Biochem 1, but we were talking about regulation. We were talking in general about positive and negative allosteric regulation. It was like the very first time we talked about it. I know, like, can you believe that? Forever ago. And there was a great animation on there's always a great animation on ATC case. So this is the enzyme, right? That eventually will make CTP. So the CTP is the final product, which will come back and negatively regulate the ATC case. So I'll have to go back and look and see if I can find which animation that is, because you should have access to the whole book, right? Everybody does. So it was in the very beginning. I'll have to go. But you had no idea of the context back then. All you knew was this is something that binds to, changes the enzymatic pocket, prevents the enzyme from functioning, right? That was the story that you were following then. But now what we're looking at here is we know how all these things are made. So this ATC case is the enzyme that converts carbamyl phosphate to carbamyl aspartate. So we can use carbamyl phosphate in the urea cycle, right? So you don't want to stop this process from happening, because then you'll interfere with more than just nucleotide biosynthesis, right? So we want to regulate the first step that is important in the nucleotide biosynthesis pathway and not the urea pathway, right? So that's bacteria, pretty simple and easy. Your final end product is a negative inhibitor, and then your starting reagents are positive effectors. Pretty easy. Animals, guess what? A little more complicated, right? So in this case, we are going to regulate this step between before upstream of the carbamyl phosphate. And the reason that we want to do this is that we have to think about what substrates we need in order to make it go. For us, we're starting with PRPP, right? Do we use PRPP for anything else? No, we use glutamine, we use bicarb, sure, we use ATP, absolutely, but we don't use PRPP. So PRPP is going to be a great positive effector of this whole pathway. We also want our final end product to be a what? Negative inhibitor, absolutely, right? Okay, so now we gotta look at some of our other inhibitors. CTP will come back and inhibit CTP synthetase. But why would GTP be a positive effector? We haven't talked about it yet. I know I want my end product to come back and inhibit the previous reaction, right? Then I'll end up with more UTP, right? But why would I want GTP to be a positive effector? Because C is the complement of G. So if you have lots of G, we want to make an equal amount of C, right? They have to be base paired. They absolutely have to be. And then you also want UTP and UDP, these products, to come back and inhibit the initial reactions. So that's another just example of feedback inhibition. Okay. Okay. Then the last place that we have regulation, right here. So UMP here. UMP is going to come back and inhibit the addition of PRPP to the orotate monophosphate, right? Because we could use the PRPP for something else if we have enough of the UMP made. You could go in and make your purines, right? So you want to be able to use it for other things instead of your pyrimidines. Okay, pretty good. What you got? Yeah, that's at the beginning.
I was thinking about how you said
Compliments. Mm-hmm. Mm-hmm.
I was like, well, that's
the whole point.
Starting.
All right, so we talked about purine degradation. We need to talk about pyrimidine degradation, right? So if you kind of look at your overall pathway that's happening, right? Your CMP and your UMP are basically going to enter the same pathway. Your DTP is going to be different, right? So your CMP and your UMP, those are the two that are structurally more similar to one another. You're going to go to uridine to uracil, right? Uracil is just the individual base. Then you go and dehydrate, right? So you got dihydrouracil, and then we're going to come on down, and we're basically going to make alanine. So now we can use this, right? Beta alanine, we can use that in the cell. That's going to be a good substrate. Our DTPs, our TN, oh my gosh, if I can't speak today. The TMPs, ooh, those are going to be broken down, and eventually we're going to make amidobutyric down here. So this one has a few other sources, few other things that it can be used for the cell, but you don't have to know a whole lot about that. They're basically converted to coenzyme A. Either one of these can go to CoA and be used in metabolism pathways, right? So they're used in many other things as you go along. They're not going to be degraded because they don't have the same structure, so you can't turn them into uric acid. It's going to be a completely different pathway. All right, there's one last thing I want to talk about today. So do you remember when we were talking about cancer, right? Very well, right? Okay, so what do we give patients to treat them for cancer? We give them chemotherapy, right? We give them chemo and radiation, right? So radiation is completely and utterly non-specific. Pretty terrible, pretty hard on the body. But we're going to talk about a chemotherapy reagent, and that's 5-fluorouracil. So 5-fluorouracil inhibits thymidylate synthesis. So we're going to inhibit basically DNA synthesis, right? DNA. There are some patients who were given this drug in the very beginnings of chemotherapy. And these patients were given what they thought was a normal level of this drug, and it killed them. And other patients didn't die. And so the question was, why did you have this really small subset of population that you gave what you thought was the correct dose of this drug, right? This is the same dose that you gave to everybody else. And these patients were dying at a crazy, crazy rate. So this inhibits thymidylate synthesis. So what reaction are we inhibiting? We're inhibiting the DUMP conversion to DTMP, because we know we need to have it in the 3-phosphate, right? So if you don't have this, you're not going to be able to synthesize DNA. You're not going to be able to incorporate your TMPs into DNA, RNA, whatever, it doesn't matter. So normally, right, if you take 5-fluorouracil and you give it to a patient, most of it is going to be degraded in the body. So 80% of the drug is degraded in the body. And so 80% of it goes this way and gets degraded as an inactive drug in the body. So you give someone a dose, and the dose that you're actually giving them is only 20%. That 20% will go and kill the cancer cells, right? But you have some people who have a deficiency in the enzyme that will actually break down 5-fluorouracil into a non-toxic byproduct. So instead of giving them 20%, you are giving them 80%. Nope, 100%. So 20, 40, 60, 80, 100, 4 times the dose of a toxic, I mean, it's a toxin, it is. So these patients get 4x dose, right? It's not, the dose didn't actually change. You gave them the same amount of the drug, but because they're lacking the enzyme, the dihydropyrimidine dehydrogenase that will convert it and degrade it and make it non-toxic, you're giving them all of it. And so those patients had severe toxicity and they died.
So it wasn't the doctor's fault.
It wasn't. They prescribed the right amount.
It was just the deficient enzyme.
It was that they had a mutation already. And that enzyme that broke down the drug to make 80% of it non-toxic didn't work. And so now, if you're going to be given chemotherapy, they screen you if they plan to use this drug to make sure that that never happens again, right? You think you're helping somebody, you think you're treating somebody, and then they die. Well, the same thing happened with the SCID people. I mean, you know, yes, it's absolutely awful, but we're learning from it, right? So now we know to screen patients. So pretty interesting, All right, I'm stopping there. We're pretty close to time. All right. But I thought that was a nice, bring it back around to what we started with in the beginning. Any questions? It all leads back to cancer. I need to go and look for that animation, huh? I will see if I can find that animation. Tell you which one, at least email y'all and tell y'all which one. Make sure your exams go back on the table. I hope y'all have a great Thursday. I will have all the rest of the case studies graded and posted. My goal is by Sunday night to have everything else graded and posted. Yep.